Hi, my name is Sohail and today I am going to discuss the models of DNA replication and mechanism of DNA replication. In molecular biology, DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication is the process by which DNA makes a copy of itself during cell division. There are three models of DNA replication. Number one is conservative model, number two is semi-conservative model, and number three is dispersive model. In conservative model, both strands of parental duplex would remain intact, so they are conserved, and a new DNA molecule is synthesized. So, the parental molecule, it directs the synthesis of an entirely new double-stranded DNA molecule. In semi-conservative model, one strand of parental duplex remains intact in the daughter strands, so they are semi-conserved, and a new complementary strand is synthesized. So, daughter molecules would consist of one parental strand and one newly synthesized strand. So, after one round of replication, each DNA molecule has one old strand and one new strand. In dispersive model, DNA replication results in two DNA molecules that are mixtures or hybrids of parental and daughter DNA. So, in this model, each individual strand is a patchwork of original and new DNA. In dispersive model, the parental molecule is distributed more or less randomly between two daughter molecules. The semi-conservative model is intuitively appealing model because separation of two strands provides the two templates, and each of which carries all the information of the original molecule. It also turns out to be correct one. Semi-conservative DNA replication by base pairing appears simple and straightforward. The actual process is highly regulated and requires a replication machine containing many types of protein and enzyme molecules. In eukaryotes, 88 genes are involved in DNA replication. DNA replication is divided into four steps. Number one is initiation phase, number two is extension or polymerization phase, number three is mechanism of DNA polymerase III activity, and number four is termination phase. DNA replication begins at specific sites on the DNA molecule that is called origins of replication, where small sections of double helix unwind. DNA helicases are helix destabilizing enzymes that bind to the DNA at the origin of replication and break hydrogen bonds, thereby separating the two strands. To understand the DNA replication, we need to understand how the bases in DNA are composed. DNA contains four types of nucleotides, that is adenine, guanine, cytosine and thymine. There are two hydrogen bonds between adenine and thymine and three hydrogen bonds between guanine and cytosine. So, the double stranded DNA molecule cannot split into two strands while it is still helical. Therefore, helicase enzyme that causes the unwinding of DNA, at the same time it separates the strand. So, this is the enzyme that is called helicase enzyme, and this helicase enzyme is responsible for unwinding of DNA molecule. At the same time, that is also responsible for the breaking of hydrogen bonds between adenine and thymine and guanine and cytosine, so that two strands they get separate from each other, and each strand that act as a template strand. Now, both DNA strands they replicate at the same time at the junction between the separated strands, which is Y-shaped structure that is called replication fork. So, this is the point which is known as replication fork, where DNA synthesis takes place. Energy from ATP is used to help move the helicase enzyme along the DNA molecule and breaking the hydrogen bonds between the nitrogenous bases and parting the two strands. Once the DNA helicases that separates the strands of DNA, then there is a protein that is called single strand binding protein. So this is the single strand binding protein that binds to single DNA strand and it stabilizes them, which prevents the double helix from reforming until the strand are replicated. Now single strand binding protein that is not only responsible to stabilize the strands, that is also responsible to prevent the hydrolysis by the action of different enzymes. Enzymes called topoisomerases that produce breaks in the DNA molecule and rejoin the strands, relieving strain and effectively preventing the supercoiling and knot formation during replication process. So this is the enzyme which is called topoisomerase enzyme, and this enzyme is responsible to relieve strain during DNA replication. There are two ways that topoisomerases reduce supercoiling. Some topoisomerases, they produce a temporary break in polynucleotide backbone of a single strand of DNA and pass that strand through excessively twisted part and then reseal the break. Other topoisomerases, they break both DNA strands, pass some of the helix between the cut ends and then reseal the break. Topoisomerases that gives the DNA more relaxed configuration. DNA gyrase is the topoisomerase involved in DNA replication. The enzymes that catalyze the linking of successive nucleotide subunits are called DNA polymerase enzyme. So this is a DNA polymerase enzyme that is involved in DNA replication, and it is responsible to link the nucleotides, and this linking is specific, as we know that adenine is bonded with thymine and guanine nucleotide is bonded with cytosine nucleotides. DNA polymerase enzyme add nucleotides only to 3' end of a growing polynucleotide strand, and this strand must be paired with DNA template strand. So this is the template strand and this is newly synthesized strand. A newly synthesized strand that is called a leading strand if it is growing towards the replication fork, and lagging strand if it is growing away from the replication fork, because the lagging strand is synthesized in small fragments, and these fragments are called Okazaki fragments, and the direction of synthesis is from 5' to 3' in leading strand as well as in the lagging strand. In DNA molecule, the two strands are anti-parallel. One strand is directed from 5' to 3' and second strand is 3' to 5'. So these two strands are anti-parallel to each other, and DNA can only be synthesized in one direction, and that is 5' to 3'. So one strand, that is leading strand, it is growing towards the replication fork, and the second strand, which is called lagging strand, that is growing away from the replication fork. And the lagging strand is synthesized in small fragments with periodic starts and stop, and they are synthesized away from the replication fork, but the direction is 5' to 3'. Replication of lagging strand is therefore made in segments, and these segments are called Okazaki fragments, after Japanese scientist Riji Okazaki, who discovered them. DNA polymerases, they add nucleotides only to 3' end of existing polynucleotide strand. Now there is a question, then how is the DNA synthesis initiated once the two strands are separated? To initiate the synthesis of new strand, a short piece of RNA is synthesized by an enzyme which is called DNA primase enzyme. And this DNA primase enzyme, it synthesizes a short piece of RNA that consists of 5 to 14 nucleotides, and that is called RNA primer. So RNA primer is synthesized by DNA primase enzyme, and at that point where replication begins. So DNA replication begins with the synthesis of RNA primer with the help of this enzyme, which is called DNA primase enzyme. After a few nucleotides have been added, now the DNA polymerase enzyme that displaces the primase enzyme and subsequently adds subunits to the 3' end of this short RNA primer. DNA replication is discontinuous in one strand and continuous in other strand. In DNA, the two strands are anti-parallel, meaning that one strand runs in 3' to 5' and second strand runs from 5' to 3'. This means that one strand can be synthesized in continuous fashion from an initial RNA primer, but the other strand must be synthesized in discontinuous fashion with multiple priming events and short sections of DNA being assembled. As we know that DNA replication begins at the origin of replication, and two strands replicate at the same time at replication fork. The position of replication fork is constantly moving as the replication proceeds. So two identical DNA polymerase molecules, they catalyze replication. So one of these adds nucleotides to the 3' end of new strand that is always growing toward the replication fork, and this strand is leading strand. Because this strand is synthesized smoothly and continuously, so it is called leading strand. Lagging strand is synthesized in small fragments, and these fragments are called Okazaki fragments. And Okazaki fragments that has 100 to 2000 nucleotides. And these Okazaki fragments were first of all discovered by Japanese molecular biologist Riji Okazaki. So DNA primase enzyme that periodically catalyze the synthesis of new RNA primer as the replication fork proceeds, and then new Okazaki fragment is synthesized in case of lagging strand. When the RNA primer of previously synthesized fragment is reached, then DNA polymerase enzyme that degrades and replaces the primer with DNA. The fragments are then joined by an enzyme which is called DNA ligase enzyme. DNA ligase enzyme links the 3' hydroxyl group of one Okazaki fragment to the 5' phosphate of the DNA immediately next to it by forming a phosphodiester linkage. Let's watch this simulation.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Let's watch this slideshow.